Welcome to the debate. If you were to count every single biological entity in your gut right now, you would find a massive, sprawling, an almost incomprehensibly dense ecosystem.
Right. It is literally teeming with life.
Exactly. But here is the paradox. The bacteria that we have spent decades meticulously studying, cataloging, and basically treating as the undeniable rulers of that ecosystem, they are actually outnumbered.
Heavily outnumbered.
Heavily, 10 to 1, by a hidden world of viruses. So today, we're asking a fundamental question. Have we been looking at the wrong microbes this whole time? Has our understanding of the human microbiome been fundamentally skewed by what we might call a bacteriocentric dogma?
I love that term, bacteriocentric dogma.
It fits perfectly. We are diving into a curated collection of peer-reviewed papers, clinical analyses, and reviews published between 2021 and 2026. And this research points toward a major paradigm shift, moving away from just looking at bacteria and toward a multi-kingdom pan-microbiome. I will be defending the perspective that bacteria, primarily due to their massive physical footprint and just their sheer metabolic output, remain the indispensable functional core. They are the true bedrock of human health and host microbe interactions.
And I will be presenting the contrasting perspective today. I argue that the largely overlooked non-bacterial dark matter of the microbiome, so specifically the virome, the fungi that make up the microbiome, and the archaeome, are actually the master architects of this ecosystem.
The master architects, even with their tiny biomass?
Even with their tiny biomass, yes, because they exert a disproportionate top-down ecological control. Without them, you do not have stability, you don't have proper human immunity, and, well, you ultimately cannot understand disease progression.
All right, well, I want to start by looking at the sheer undeniable scale of the bacterial bedrock, because you simply cannot ignore the mathematics of the human gastrointestinal tract here. Recent highly accurate volumetric counts have shown that a standard human harbors roughly 38 trillion bacterial cells.
It's a staggering number. Yeah.
It's huge. That is a true one-to-one ratio with our own human cells. But it isn't just about them passively taking up space, you know. It is about the heavy lifting they do for our survival. I mean, the human genome is actually quite limited. We completely lack the genetic tools to break down complex plant fibers.
Right, we can't digest our own food, essentially.
Exactly. We rely entirely on bacterial enzymes, cazymes, to ferment those non-digestible carbohydrates. And the byproduct of that bacterial fermentation is a constant stream of short-chain fatty acids. Molecules like butyrate.
The classic gut fuel.
Yes, and butyrate isn't just waste. It is literally the principal fuel source for the cells lining our colon. It provides the energy to keep our intestinal barrier sealed tight, preventing a leaky gut, and actively calms our local immune system.
I mean, the metabolic output is massive. I completely agree with you there. Bacteria are the factory workers churning out the product.
They are the foundation of our homeostasis. And what makes this bacterial bedrock so incredibly powerful is the concept of functional redundancy. The gut is packed with hundreds of different bacterial species. This means you have multiple coexisting, genetically completely different bacteria that can perform the exact same job.
So if one goes down,
Exactly. It acts as a massive biological shock absorber. If a sudden change in diet or a passing stomach bug wipes out one specific type of bacteria, the ecosystem doesn't just collapse. Another type of bacteria simply steps up, turns on its identical metabolic pathways, and keeps churning out that essential butyrate.
sure, I see the logic.
This is exactly why the vast majority of our clinically validated therapies right now, from highly defined live biotherapeutics to your traditional probiotics, they rely entirely on bacterial mechanisms. The bedrock is resilient, it heals itself, and it drives our biology.
Well, I look at that exact same ecosystem, but I come at it from a completely different angle. Because focusing solely on physical biomass and, who is producing most enzymes, that is a reductionist error.
A reductionist error.
It is, and it is an error that we fell into because of historical technological bias. For decades, the standard way we analyze the microbiome, was through something called 16S sequencing.
Right, looking at the ribosomes.
Exactly. It worked by looking for a specific genetic marker that only exists in bacteria. So intrinsically, our early tools were entirely blind to viruses, blind to fungi, and largely blind to archaea. It is the classic streetlight effect, right?
We built a bacteriocentric dogma because we only looked where the light was shining.
Precisely. But over the last few years, as we have shifted to advanced shotgun metagenomics, which sequences all the DNA in a sample regardless of what kingdom it belongs to, we have hit this viral bacterial paradox.
Which brings us back to the numbers game, just a different set of numbers.
Yeah, exactly. With viruses, which in the gut are mostly bacteriophages, so viruses that specifically infects bacteria, account for less than 0.1% of the total microbial weight because they are so incredibly minuscule. Yet, they are the most numerically abundant biological entities inside us.
Wow, okay.
In the gut, virus-like particles outnumber bacterial cells by a ratio of 10 to 1. In some of the mucosal layers lining the intestine, it is 20 to 1. So while your bacteria might be the factory workers synthesizing the metabolites, the virome, the microbiome, and the archaeobiome are acting as the management.
The management.
Yes, they are top-down population regulators. They are thermodynamic sinks. They are the high-level calibrators of human immunity. So my position is that without these non-bacterial architects, that bacterial bedrock you just described, it isn't a resilient biological buffer at all. It is an ecologically fragile, unstable monolith that would just tear itself apart.
Okay, I see why you think that, but let me challenge this idea of ecological control versus functional resilience. You are suggesting the virome is managing the system, but think about my earlier point regarding functional redundancy.
The shock absorber idea.
Right. That bacterial ecosystem functions like a massive, decentralized power grid. If a localized storm takes down one substation, representing one specific bacterial strain, the grid does not fail. Power is automatically rerouted through dozens of other substations performing the exact same function. The bacterial network is robustly self-healing. So my question to you is, how can transient, non-metabolizing viruses, entities that don't even possess their own cellular machinery, claim to regulate a system that is so heavily buffered and entirely self-sufficient?
Because I would argue that your bacterial power grid only remains decentralized and diverse because of those bacteriophages.
Wait, how does that work?
Well, if you actually leave a bacterial community to its own devices without any predatory regulation from viruses, it doesn't stay perfectly balanced. One highly efficient, aggressive bacterial strain will naturally outcompete all the others. It will monopolize the nutrients, starve out the competition, and suddenly your beautifully diverse, decentralized power grid turns into a single, highly vulnerable, centralized power plant.
So you're saying the system naturally tends toward a monopoly?
It absolutely does. And phages prevent this through a dynamic known as kill the winner.
Kill the winner. That sounds aggressive.
It is. These lytic viruses are programmed to specifically seek out, infect, and rapidly destroy whichever bacterial strain becomes the most dominant. By constantly knocking down the most abundant taxa, they prevent any single bacterium from monopolizing the environment. They force the ecosystem to maintain that high diversity.
Okay, I see.
So that functional redundancy you are praising, the bacteria can't maintain it on their own. The viruses enforce it.
The destruction of the bacteria is what maintains the diversity of the bacteria. It is a fascinating ecological dynamic, but surely that is just a localized skirmish.
Oh, it is much more sophisticated than just a skirmish, especially when the ecosystem is under stress. Viruses don't just kill, they also use a strategy called lysogenic symbiosis.
Right, where they integrate into the genome.
Exactly. Instead of destroying the bacterium, the virus splices its own genetic material directly into the bacterial host's DNA. It hides there. We call this piggybacking the winner. And these hidden viral genes actually give the bacteria new survival traits and metabolic abilities when the environment gets harsh. We see this play out constantly in the clinical reality of bacterial vaginosis, or BV.
Right, which is a massive clinical frustration. Because when we treat BV with broad-spectrum antibiotics, we absolutely decimate those bacteria, but the recurrence rate is devastatingly high. It comes back in something like 80% of cases within a matter of months.
Exactly. And the virome explains why our bacterial treatments fail here. When you give that antibiotic, you clear the bacterial cells out. But those lysogenic viruses, the ones that integrated into the bacteria,
They get released?
they get released. And they remain in the environment as floating genetic reservoirs. They literally stabilize the structural biofilms left behind. As soon as the antibiotic pressure is gone, these viral reservoirs dictate the bacterial comeback.
Wow.
They rapidly integrate into new colonizing bacteria, passing on the genetic instructions to rebuild the exact same pathogenic state. The bacteria are just the workers returning to the site. The viruses are literally holding the architectural blueprint of the disease.
That is a very compelling way to look at population control. But let me pivot here, because controlling which bacteria grow is still just local microbiology. It's microbes fighting microbes.
Fair enough.
If we want to talk about true human health, we have to look at how these microbes directly interact with our own human biology. We have to look at the immune system. And this is where your argument for the non-bacterial side, particularly the fungal side, the microbiome, runs into a massive mathematical problem for me.
Okay, let's hear the math.
We both acknowledge fungi are present in the gut, but let's be precise about the footprint here. Fungi only comprise about 100,000 to a million cells per gram of feces. That is roughly 1/10 of 1% of the total microbial biomass.
Right.
They are dwarfed on an astronomical scale by bacteria. Our human immune cells are constantly bathed in a chemical deluge of bacterial short-chain fatty acids, secondary bile acids, and bacterial vitamins. So how can a kingdom with such an infinitesimally small footprint competitively influence our immune system against the roaring tide of bacterial metabolism?
Because biomass does not equate to biological impact. We are talking about entirely different domains of life. Fungi are eukaryotes, like us. They compensate for their low numbers with immense structural and chemical complexity.
Okay, but how does that bypass the math?
Look at a bacterial cell wall, it's relatively simple. But a fungal cell wall is a highly complex, dense fortress made of unique sugars, specifically beta-glucans, chitin, and mannans. These are incredibly potent alarm signals for the human body. Our innate immune system has evolved over millions of years to be exquisitely paranoid about fungi.
Paranoid, right.
Very much so. The human guard cells sitting just below the intestinal lining, like macrophages, are absolutely covered in a specific sensor called dectin-1.
Right, and dectin-1 is purpose-built to recognize those fungal beta-glucans.
Precisely. And when a tiny fungal sugar binds to that dectin-1 sensor, it doesn't just cause a localized inflammatory blip. It flips a master switch inside the human immune cell. It triggers a cascade that literally reaches into the immune cell's DNA, unspools certain sections, and changes how the DNA is folded.
Epigenetic reprogramming.
Yes, epigenetic reprogramming. Because of this structural change, the immune cell's entire baseline metabolism speeds up. This process is called trained immunity. That immune cell is now permanently hyperreactive. It will respond more aggressively to all future threats, even completely unrelated bacterial or viral infections.
That is a massive shift from just a few fungal cells.
Exactly. So yes, the fungal population is tiny, but it acts as an immunological sentinel that permanently calibrates the baseline reactivity of your entire innate immune system.
I will certainly grant that dectin-1 signaling is a potent mechanism, but we must also acknowledge the reality of how that fungal threat is actually managed day to day. Keeping those fungi in a harmless, commensal state is actively dependent on the bacterial bedrock.
How so?
In a healthy gut, bacteria exert continuous, crushing suppressive pressure on opportunistic fungi like Candida albicans. Those bacterial short-chain fatty acids we talked about earlier, they actively chemically constrain Candida, forcing it to stay in a benign yeast form.
Sure, when the bedrock is healthy.
Right. But when we use heavy antibiotics and fracture that bacterial bedrock, we create a microbial vacuum. The Candida, which doesn't care about antibacterial drugs, rapidly changes its shape, grows invasive root-like structures, penetrates the intestinal wall, and causes severe tissue damage. The only reason that tiny fungal footprint doesn't overwhelm us is because the bacterial bedrock keeps it constantly in check.
But that inter-kingdom tug of war is exactly my point. You are focusing on the physical invasion of the fungus, but you are missing the invisible metabolic warfare happening before that.
Invisible warfare?
Yes.
Fungi and bacteria fight ruthlessly over resources, particularly over dietary amino acids like tryptophan. When fungal populations start to expand, even slightly, they are incredibly efficient at hijacking the tryptophan from the food we eat, stealing it for their own pathways.
Which starves the bacteria of the raw material they need.
Exactly. And why does that matter to the human host? Because the bacteria need that tryptophan to synthesize specific molecules called indoles. These bacterial indoles are crucial chemical messengers that travel throughout the human body. They bind to receptors that suppress inflammation, not just in the gut, but all the way up in the brain.
They cross the blood-brain barrier?
They are central to maintaining the blood-brain barrier. So when the fungi hijack the tryptophan, the bacterial indole production completely stalls out. Without those indoles, the brain's anti-inflammatory defenses drop, the blood-brain barrier gets leaky, and you get central neuroinflammation.
Oh, wow.
The fungi are actively driving neuroinflammation via the gut-brain axis, not by invading tissue, but simply by changing the metabolic calculus of the bacteria.
I hear the complexity of that trans-kingdom crosstalk, I really do. But if the non-bacterial side of the equation is as critical as you say, we would see that reflected in modern medicine. And honestly, we don't.
I disagree, but go on.
When we look at how we actually treat severe, life-threatening dysbiosis right now, the clinical evidence points right back to bacteria. Let's look at therapeutic engineering. The field is rapidly moving away from fecal microbiota transplantation, or FMT.
The shotgun approach.
Right. FMT transfers the whole ecosystem, viruses, fungi, bacteria, but it is totally undefined. It is a roll of the dice that carries the risk of transferring unrecognized pathogens. Instead, the FDA and modern medicine are moving toward highly controlled, defined bacterial consortia.
Like for C. diff.
Exactly. Take purified bacterial spores used to cure recurrent Clostridium difficile infections. We take a few dozen highly specific, laboratory-grown bacterial strains and deliver them to the patient. No fungi, no viruses, and they work beautifully.
In the short term.
They engraft, they outcompete the C. diff pathogen, and they restore the patient's health. This incredible precision proves that the bacterial bedrock alone is entirely sufficient to rebuild a broken ecosystem.
It proves they can put out a localized fire in the short term, but computational models and emerging long-term data suggest that embedding isolated bacterial strains without their native viral regulators or, without their non-bacterial metabolic partners is ecologically doomed in the long run.
Doomed? That's a strong word.
It is, but think about it this way. You are basically building a massive manufacturing plant with thousands of workers, but you are refusing to install an exhaust system.
An exhaust system? What do you mean by that?
I mean the archaeome, the most neglected kingdom of all. Archaea are single-celled organisms that look like bacteria but are fundamentally distinct. In the human gut, this kingdom is dominated by methanogens like Methanobrevibacter smithii.
Okay, the methane producers.
Right. Now, unlike your bacteria, they do not ferment carbohydrates. They act as the gut's thermodynamic exhaust system. As your bacterial bedrock is busy fermenting all that fiber, it produces massive amounts of hydrogen gas as a waste byproduct.
Sure, it's a known output.
But if that hydrogen gas is allowed to accumulate, the physical pressure inside the gut rises, and it creates a thermodynamic bottleneck. The chemical reactions essentially hit a wall. The bacterial fermentation engine literally chokes on its own exhaust and stalls out.
And the archaea are what prevent this stalling?
Exactly, through a process called hydrotrophic methanogenesis.
Which is a mouthful.
It is a mouthful, but it simply means these archaea act as scavengers. They aggressively suck up that accumulating hydrogen gas, combine it with carbon dioxide, and convert it into methane gas, which can then be safely expelled. They exist in a strict, codependent partnership with the bacteria.
I see.
If you just drop a defined bacterial consortium into a patient without ensuring that the archaeal exhaust system is functioning, that community remains highly vulnerable to completely stalling out. And we are already hitting the limits of this bacterial-only thinking in the clinic right now.
Walk me through an example of where bacterial therapeutics are failing because of this, then.
Look at irritable bowel syndrome with constipation, or IBS-C. A major recognized driver of this condition is intestinal methanogen overgrowth.
Where there's too much methane.
When you have too many of these archaea, they produce too much methane gas. That methane acts directly on the neuromuscular apparatus of the human gut, paralyzing it and severely slowing down digestion. And because archaea have completely different cell walls than bacteria, they are intrinsically immune to almost all of our broad-spectrum antibiotics.
Right, so you can't just give them a standard antibiotic.
You absolutely cannot treat this debilitating condition by manipulating bacteria. That failure has led to the development of targeted archaeabiotics, like a drug called SYN-010.
Which specifically targets the archaea.
Yes, it acts as a competitive inhibitor at a critical juncture in the archaeal methane-producing machinery. It essentially unplugs the exhaust system just enough to relieve the IBS-C without causing any collateral damage to the bacteria.
Okay, but you have to admit that is a highly targeted pharmaceutical chemical intervention. It's not an ecological replacement strategy.
Okay, then let's look at actual ecological replacement, fecal virome transplants, or FVT.
Just the viruses?
Just the viruses. Researchers are now taking donor stool, heavily filtering it to remove absolutely every single bacterial cell, leaving only the viral fraction. So predominantly bacteriophages. They are administering this sterile viral liquid to patients.
And what happens?
In robust preclinical models of inflammatory bowel disease, administering just the viruses successfully restored healthy bacterial diversity and reversed the damage to the intestinal lining.
Without adding a single bacterium.
Think about that. They are hitting the reset button on the entire regulatory framework of the gut community and driving it back into homeostasis without adding a single bacterium. It proves that manipulating the viral dark matter can control the ecosystem from the top down.
It is undeniably a fascinating frontier, I'll give you that. To summarize my position in light of our back and forth today, the complex behaviors of phages, the immune calibration by fungi, and the thermodynamic role of archaea are incredibly intricate. However, the sheer physical scale and the massive, indispensable functional output of the bacteria simply cannot be sidelined.
They are important. I don't deny that.
Bacteria remain the definitive functional core of our microbiome. They dominate host metabolic interactions. They are the ones producing the short-chain fatty acids, deconjugating our bile acids, synthesizing our vitamins, and they form the solid empirical basis of our most successful, currently validated microbiome therapies. The bedrock might require maintenance from other microbes, sure, but it is still the bedrock holding the house up.
And my summary is that treating the human gut as a monokingdom bacterial bioreactor is a fundamental ecological mistake. The bacterial bedrock operating in isolation is just an unstable monolith waiting to fracture. You cannot fully understand, and you certainly cannot permanently repair this ecosystem without acknowledging the master architects.
The management.
The management. You have the virome utilizing lytic predation to physically force bacteria to remain diverse. You have the microbiome acting as an immunological sentinel, permanently calibrating our baseline immunity by physically altering the DNA of our immune cells. And you have the archaeome, serving as the thermodynamic exhaust system that prevents the whole engine from choking on its own waste. The dark matter is entirely what holds the visible matter together.
I think where we absolutely converge today is on the historical and methodological constraints that created this blind spot in the first place. For decades, our reliance on 16S sequencing created a massive artifact in the data. Because the tool only looked for bacteria, we naturally assumed bacteria were the only things that mattered.
Completely agreed. It is really only with the advent of long-read, shotgun metagenomics, where we sequence everything in the sample indiscriminately, along with, advanced computational tools to piece together viral genomes, that we are finally pulling back the curtain on these hidden kingdoms.
The transition from a rigid bacteriocentric dogma to a true multi-kingdom pan-microbiome paradigm highlights just how immensely complex human biology is. Understanding this massive trans-kingdom crosstalk, from a virus shaping bacterial genetics to survive stress, to a fungus stealing tryptophan and causing neuroinflammation, to an archaeon pulling the thermodynamic levers, it's clearly the essential next frontier for addressing chronic, complex diseases.
It forces us to respect the ecology as a dynamic, interconnected whole, rather than just isolated bacterial parts.
Exactly. We strongly encourage all of our listeners to explore the source material further, because the more you read, the deeper this rabbit hole goes. And as we leave you today, the next time you think about your microbiome, try to look past the trillions of bacterial workers churning out metabolites. Remember to look at the hidden management layer, the viruses, fungi, and archaea, quietly pulling the ecological levers that keep the whole system from tearing itself apart.